Thanks very much, Asunta. So this presentation shows was carried out only with the participation of CICI, and you already mentioned the contributors. So background. Um, this slide shows you pictures of Alnidia merili or Manila palm, which shows different types of symptoms, but these were found to be associated with different phytoplasmas. So these species can be infected with the subgroup A and subgroup D. Then these tables are from papers where what was reported was phytoplasmas in different palm species. And as you can see, it's again A or D. And in this other table, the presence of phytoplasmas was studied in insects, Plaxius crudus. And in most cases, it is A, then D, but there are two cases of E. So, but again, it's A and D, the most abundant phytoplasmas. And this one is a third case. In this one, this study, what's done is transmission of phytoplasmas by Plaxius crudus. I'm just going to show you the results related to the subgroups. These are the subgroups in the plants where the phytoplasmas were transmitted. As you can see, it's A and D. And in the case of the insects, again, we have A and D. So the message there is that most of the phytoplasmas found in plants, but also in insects, are either subgroup A or subgroup D. This I have already shown, this information as well. Well, in order to study the presence of phytoplasmas, you take samples. These kind of pictures have been shown before. We reported this for first time in 2000. And then with those samples, you can use molecular tools for detection and identification of the subgroups. With this methodology presented here, it can take about 10 days from start to finish. It's good to have the methodology, but it takes long. So the purpose of this study was to develop a qPCR method that could be faster, easier, and less expensive for this dedicated application of detection and identification of these two abundant subgroups, and hopefully with a method that will require one reaction within a single tube. Well, results. So for this purpose, method was developed. I'm not going to present details of how it was developed, not specific information either, because Tropicsafe considered that this could be a candidate for exploitation. But in a few words, you take the sample, and then amplification takes place within the system. It can generate two different types of signals. One is a signal for group A and another one for group D. Then you can see through the monitor whether you are having the subgroup A or the subgroup D, or maybe both. So what it was done first for this, for testing this, was specificity, and these are the phytoplasmas that were tested. As you can see here in red, the only ones that could be amplified were either A or D, with one signal or the other. But if you do a standard universal nested PCR, you get amplification for all of them. Then sensitivity in both cases was the same. You can go as low as 0.01 nanograms for getting detection. Here, I'm showing you two sets of samples. In the first one, what we have is again A and B depending on related particularly with the species. So if it is coconut or Trinax radiata, it could be A, and for Phoenix canariensis, it could be B. And in these particular samples, sequencing was carried out, and the subgroups that were found through the qPCR method were also coincided with those by sequencing. This other set of samples also showed this is only coconut, that subgroup A was found. No sequences have been done here. It will be done. So if we compare in regards the time taken, as I said, what we are doing traditionally is a 10-day method. And in the case of the project new method, it takes 40 minutes and it will tell you whether it is A or B. So in conclusion, the results presented show the capacity of the new qPCR assay for detecting specifically and rapidly DNA of these two subgroups within a single tube and with a single qPCR reaction. This assay could be very useful for research and epidemiological purposes. So thanks very much.